What was the reason when you founded Cyrom that you thought IROM2 could be targeted differently this time?
The idea was to find a new way of targeting TACE, which is the molecule that is being regulated and influenced by ARM2. So the idea of founding a company on that basically came from one of our co-founders, Carl Blobel, who discovered IROM2 and just a couple of years before. And TACE used to be a very hot target in the past. So many big companies, pharma companies, tried to develop TACE inhibitors, which all failed for certain reasons. And IROM2 was discovered as a critical regulator, and that offered the opportunity to target TACE in a different way. And we found that extremely exciting because it was a novel approach. It was a new way of targeting TACE, which failed before. And we were very optimistic that this is the key of targeting TACE.
What was it that went wrong previously that you felt like you could do things differently?
Well, the inhibitors that were developed by different companies before targeted TACE itself. And one needs to know that TACE is a very central protein that is involved in the regulation of multiple pathways, many pathways that are involved in disease, but also importantly, also pathways that are critical for normal physiological processes. And all those inhibitors that were developed targeted TACE globally, which in essence means that you may have the benefit of targeting those pathways that drive disease, but at the same time, you would also target pathways that are important for normal physiology. So in essence, that broad global interference with TACE led to side effects in the clinical trials and caused them to be discontinued. And now IROM2 offered the possibility to target TACE in a more selected way.
Mm-hmm.
And that is something that was basically the key to readdress TACE.